Good afternoon, everyone. My name is Mitchell Sexton, and the paper I'll be presenting today is titled CRISPR-Cas9 mediated gene knockout of Anopheles gambiae FREP1 suppresses malaria parasite infection. And here are the authors. And I'll only be talking about one section of the paper, which mainly focuses on the results of when you knock out the FREP1 gene and how that affects the Plasmodium parasite infection of those mosquitoes. Now, just for some background information, they use CRISPR-Cas9 to generate these gene knockouts, mosquitoes. And CRISPR stands for clustered regularly interspaced short palindromic repeats. And these are repeating sequences of DNA that is derived from viral DNA because it is a prokaryotic system. And it involves a Cas9 endonuclease, which is a restriction enzyme that generates double-stranded breaks, as you see in this figure. Generates a double-stranded break. And CRISPR-Cas9 can be used as a gene editing tool by scientists. And it can be used to edit specific sequences to generate double-stranded DNA breaks, like I just said, a single-base pair substitution, and it can also add sequences to the DNA. As you can see here in the figure, this would be a case of adding a DNA sequence to the DNA that you just broke, and then your final product. So the mechanism of action for CRISPR-Cas9 involves a guide RNA and a Cas9 endonuclease. And the guide RNA consists of two components, a target-specific CRISPR RNA, which can be seen here, and that binds to the target sequence of the DNA that you're trying to alter, and then also contains a transactivating RNA which associates with the Cas9, and this is the gRNA, the guide RNA, and it guides the Cas9 to the target sequence, and that is usually viral DNA or the target gene. So the mechanism of action is there's two requirements for the binding. The first one is the recognition of the protospacer, and that's just the target sequence, which can be seen right here by the gRNA, and also recognition of the protospacer adjacent motif, which is right here. Usually it has one random nucleotide followed by two guanines, and it distinguishes bacterial from foreign DNA, and that is where the Cas9 binds. And so with this all together, it can make this cut right here at the end of this target sequence. So when CRISPR-Cas9 makes a break in the DNA can be repaired by two different pathways. You have non-homologous end joining or homology-directed repair. And non-homologous end joining is error-prone, involves a lot of insertion and deletions. So for example, here would be an insertion after a DNA cut, or it can be a deletion, which in this case you see you have 4 black nucleotides that are getting rid of. Or it's homology-directed repair, which is less efficient yet more precise, so it can repair it with the correct base pair. So there's several applications of CRISPR-Cas9. It's used to knock out genes, such as it could be used to knock out induced muscular dystrophy. And the limitations is it can tolerate mismatches. Inserting DNA sequences is challenging, and non-homologous end joining is preferred over HCR, which can be kind of a problem in other research. So in this paper, why focus on malaria is the big question here, and the reason is because it's the cause of 1 million deaths per year, and 90% of those deaths are in sub-Saharan Africa, and it's caused by the protozoan parasites like Plasmodium. It's caused by 5 parasites, which can be listed right here: the falciparum, vivax, ovale, malaria, and nulci. And it's carried by female Anopheles mosquitoes, and for them to get the parasite, it requires a blood meal for egg development of that parasite. So the transmission is cyclical, so it has 5 main steps to the cycle, which involves parasite grows and multiplies in human liver cells first. The parasite invades the bloodstream and grows inside the human host's red blood cells. And then the mosquito ingests parasite during blood feeding, and then the parasite multiplies in the gut, and the sporozoite, which is the parasite, migrates to the salivary glands, and when the mosquito goes to another host, it injects the sporozoite to the human host upon another blood feeding. So here's just a picture showing the cycle. This would be the liver part, going from the liver to the erythrocytes to the mosquito, ingesting the parasite, and then it multiplying in the gut, and then the parasite here, and this oocyst traveling to the salivary glands, and then it just repeats. So the treatments and symptoms for malaria: the symptoms involve pain in the abdomen or muscles, chills, fatigue, fever, night sweats, nausea, or vomiting. And treatments just involve antiparasites and antibiotics for the infection. So the question is, they focus on FREP1, and why would they want to focus on FREP1 is the big question. So FREP1 belongs to the fibrinogen-related protein family, and it specifically stands for fibrinogen-related protein 1. Fibrinogen is a soluble protein in the blood plasma, and it aids in blood clotting by being converted to fibrin. Now, in the mosquito, it facilitates plasmodium invasion, so it binds to the gametocytes, ooconites, which is a motile zygote, and it brings them to the midgut, from there they travel to the salivary glands. So the purpose of this paper is to observe the effect that the Plasmodium falciparum infection in FREP1 gene knockout mosquitoes by using CRISPR, by breaking up that gene, the DNA in the gene, and seeing what happens from there. So before I talk about my section, previously they generated the FREP1 knockouts, and so what they did here is they created 3 gRNA expressing vectors, which can be seen right here, and they were under control of the U6 polymerase III promoter, and they cloned these into plasmid modules, and this plasmid module contained a reporter gene, which is 3 by P3 TFP reporter, and that's how they're able to detect which mosquitoes got the plasmid, because that reporter gene expresses a protein that fluoresces. And what they did is they microinjected these into embryos, and they selected for homozygous deletion mutants for the section that I'm going to talk about. So first what they did is they fed the mosquitoes on falciparum gametocyte cultures, and they fed the FREP1 mutants on these cultures along with the control groups X1 Cas9, FREP1 gRNA, and they did this through artificial membranes at 37 degrees Celsius for this, and they also fed mosquitoes, they also allowed them to feed on the Webster mice at 19 degrees Celsius for another way of just mixing up their research. So before feeding, they starved the adult mosquitoes prior, and they starved them for 3 to 5 hours. And this way, it would ensure engorgement, so no mosquitoes would just be like half fed. Unfed mosquitoes were removed, and the fed ones were incubated for 8 days at 27 degrees Celsius for the falciparum, and then 13 days at 19 degrees Celsius for the Plasmodium viridii. The midguts and salivary glands were then randomly selected, and they were studied, and then they generated dot plots using that just to give a visual of the results. So to begin with, what they found was there was a lower permissiveness to the pre-oocyst stage for P. falciparum, and the blood that they used had a very high gametocytemia, which was 0.1%, which is not exhibited in nature at all. And the wild type exhibited 98.1% infection, and the prevalence of infected mosquitoes decreased by 22% when that was compared to the X1 wild type. And the median infection intensity was decreased by 81.3% as compared to the X1 wild type as well. So here's the figures that they have in the paper. This is basically showing how, just giving a visual of the results of what is going on in the paper. So you can see here the FREP1 knockouts, very low oocysts in the midgut compared to the controls. So in the second part, what they did is they fed the mosquitoes on a 10-fold lower gametocytemia to exhibit the results. And mutants exhibited strong parasite suppression once again, had a median oocyst count of 0 in the mutant and 2.5 in the wild type. The FREP1 knockout mutants exhibited 41.3% infection prevalence as compared to the control X1 mosquitoes, which is 82.9% infection prevalence. And so this is the dot plot that they generated from that. As you see, the median is the red line here, and this can be at 0. So as you see, the FREP1 knockout is working with reducing the parasite infection. So they also want to look at the salivary glands, because they just looked at the midguts, and they found there was a decrease in sporozoite stage parasites in the salivary gland as well. Median sporozoite count of 0, and there was a 1.9-fold decrease in the FREP1 knockout mosquitoes. So it's 82.1% in the control mosquitoes, while there was a 42.9% in the FREP1 knockouts. So here's the graphs that they generated for the salivary glands for the sporozoite. As you see here, once again, significant reduction with the FREP1 knockouts as well. So basically, what we can concur from this paper is that there's significantly reduced Plasmodium falciparum infection mosquitoes, and this reduces the transmission of the parasite. But the problem is that there is adverse physical side effects such as diminished feeding propensity, slower pre-adult development, and decreased longevity. So with this said, for future research, they need to look into plasmodium-resistant mosquitoes, competition against the wild-type mosquitoes, let's say if they were released those into nature. And they also should do tests on other factors influencing plasmodium resistance, maybe other genes, and so forth. And here are my references.